Donc je suis très content de vous introduire Zoher Gueroui. Il me rappelait à midi qu'on s'est connus quand j'étais à Strasbourg et j'allais faire un cours de master à Lyon. Et il assistait au cours et après l'après-midi, je n'avais pas grand-chose à faire à Lyon, donc j'allais dans son labo. Donc j'ai passé 3 ou 4 semaines comme ça, tous les jours dans son labo. Ils avaient une manip très belle avec Bruno Berge sur les ARN polymérases qui sautaient au-dessus de trous. Je me souviens encore de ça. Je ne me souviens pas de la suite, mais je me souviens de ça. Donc après, il a fait sa thèse à Lyon. Donc, il a été postdoc à Rockefeller. C'est un enfant d'Albert Lipschaber, de 2 façons d'ailleurs, par Bruno Berge et directement. Des choses dont je me souviens, c'est que tu étais venu me voir une fois pour me raconter ces expériences que tu avais fait avec Kapoor, où tu mettais des faisceaux mitotiques et tu en mettais plein en parallèle et tu regardais la taille des faisceaux mitotiques. Chose que, si je me souviens bien, tu as continué quand tu as été à Rennes et puis même peut-être au début quand tu as été à Paris. Donc il a eu un poste au CNRS à Rennes et puis il est revenu il y a quelques années, ça doit faire 10 maintenant, non ?
Exactement.
À l'école normale supérieure à Paris, où il est au département de chimie. Alors si on regarde son site web, pas très développé, mais il y a quand même un thème général des recherches. Contrôle des protéines dans le temps et dans l'espace, dans les cellules vivantes. Donc c'est ça le thème général de ses recherches, qui correspond très bien à ce qu'il fait. Et dans ce cadre-là, il s'est intéressé aux condensats biologiques. Donc, il va nous parler de ce qu'il a fait là-dessus. Et le thème, c'est « Engineering phase separation in cells to study RNA-Protein condensate ». À toi.
Good afternoon, everyone. So it's a pleasure to be here. Thanks a lot, Jean-François, to give me this opportunity to present our work. So we are actually a small group of physical chemists, and we are developing in the Department of Chemistry at ENS, next door, some molecular tools in order to engineer phase separation, to trigger phase separations in living cells. And for that, actually, we built what we called artificial membraneless compartment or artificial condensate here, what you can see here. This is a nucleus here, and we can see these large bodies. And I will try to show you and convince you how actually there's a small artificial compartment can be used to examine some fundamental and basic questions in cell biology, in particular the role of RNA in controlling actually the formation of these granules. So as a physical chemist, it's always fascinating to see how the assembly of living systems results from the interaction of biomolecules over length scale that can cross several orders of magnitude from the nanometer scales up to the cellular scales. And actually at the intermediate scales, so typically around the micrometer, there is mounting interest in the studies of biomolecular condensates. Typically since 10 years, there is a lot of studies, and the current challenges that the field is facing is try to decipher and characterize the biochemical composition of these condensates, as well as their biophysical properties, and try to link these characteristics to their putative functions and dysfunctions. So to illustrate the diversity of RNA-containing condensate, I'm showing you here 3 different examples. So here on the top left, we can see GEMS granules in C. elegans. So in green here, it's a GFP label of a protein, an RNA binding protein called PIGL. So there are, so the PIGL granules, here you will see that they start to accumulate asymmetrically during the first asymmetric cell divisions. And these PIGL granules actually can concentrate a lot of different RNA. And their localization are very important to define cell fate, especially to define the germ cells. In a totally different context here on the right, I'm going to show you a movie where we apply a chemical, a small chemical, such as arsenite, and we can trigger actually the formations of stress granules that also contain a lot of messenger RNA, microRNA. And their putative function is that the formation of such condensates in the cytoplasm allow actually the RNA to be protected upon stress applications. What is also interesting here is that when we remove the stress granules disappear. And finally, I'm showing you here at the bottom a last example where we can see there's yellow dots here. There are RNA transport granules that can be powered by molecular motors along microtubule tracks within axons. And their functions actually are to transport locally messenger RNA, especially at the vicinity of synapse, in order to locally allow the protein production that is important to maintain memory. So the typical scale is 500 nanometers, and so there is a bidirectional transport. And what is interesting, it seems that the RNA within the condensate is translationally repressed. So there is no protein production during transport. So you can see here 3 different examples of RNA containing condensate in 3 kind of different contexts. So they have obviously different biochemical compositions, different functions. Yet, several years ago, the concept of phase transitions in cells has been proposed in order to explain potential mechanisms of biogenesis of such organelles in cells. So phase transition that is ubiquitous in non-living systems, such as this example here, where we can observe the formation of clouds that results from the condensation of water molecule at a specific temperature and pressure. And really to illustrate how these ideas has been proposed, I'm going just to remind what I found 2 very important papers in the field that has been proposed. The first one is an experiment that has been performed by a team of biochemists, the team of Mike Rosens. And the idea was very simple. They produce actually protein that can interact by low affinity and by multivalent fashion. So they produce these multimeric proteins here. And what they show in a test tube is that above a critical concentration of these proteins and above a critical valency, here 4, you can observe the formation of droplets, dense droplets here, of the proteins that are in dynamical equilibrium with a dilute phase. And so it reminds us actually the liquid-liquid phase separation that we can observe in soft matter. And the second example I would like to remind you is this landmark studies from Clifford Brangwain in Tony Hyman lab. And here I show you a movie where we apply a shear stress, some force, on the piggle granules, so the germs granule I introduced previously. And we can see actually that the germs granules are deformed under stress, and they can drip, they can fuse. And this also reminds us what we know from soft matter physics. So there's 2 examples, the fact that there is a sort of phase diagram at dynamical equilibrium, and the fact that there is some viscoelastic properties of this object allow them actually to propose.
If you wait very long, this
yes?
Does it like this, or does it separate?
Actually, the sediment on the surface. And this may preclude coalescence, and you can observe some Ostwald rippling, actually.
And in the end, you have 2 phases.
It's unclear. It has not been described here. Yeah, exactly. But indeed, this example actually that has been performed here has been reproduced in a lot of different systems, and in some systems there is some case where there is this macroscopic demixing. You observe this very often here through sedimentation in a test tube. Very often, if you wait 48 hours, for instance, you will have 2 phases. And yeah, so the global idea here was that there's biomolecules that can be made of RNA-binding protein, for instance, and RNA can interact locally, transiently, by multivalent and weak interaction to form this condensed phase here that can have different physical properties, liquid, gel, for instance. And what is interesting is that depending on the protein, this intermediate actually phase may evolve towards something more complex or different. For example, it may trigger the formation of irreversible bondings or aggregations, and you can observe, for instance, aggregates. So this is a working hypothesis, and I will, at the end of my talk, discuss this aspect. The kind of questions we are interested in the field or in our lab is to try to understand how cells, living cells, can control phase transition. For example, to localize the biogenesis of such RNA-containing granules in space. So we had actually in the last 2 lectures from Jean-François some ideas about how size control can emerge if you, for example, introduce out of equilibrium reactions. And I will tell you more about a system that we established in the lab to try to understand how biomolecular condensates can have a finite size. Something that is also very important is try to link the biophysical properties of such condensates with their biochemical functions, for example, RNA translations. And finally, one important question is try to see how actually liquid-like condensate could evolve and age towards something different, such as a gel or solid condensate. So the majority of the studies that has been performed in the past years were done in test tubes. So you may remember the talk from Simon Alberti last week, where he really deciphered the minimum requirement to form a biomolecular condensate by purifying specific effectors. So these studies actually are very helpful to really map the phase diagram and to find the minimum requirements. Yet they lack actually the complexity of working within cells. For instance, the fact that you will have promiscuity of interactions, molecular crowdings, or the fact that the cellular interior can display some heterogeneity, such as the presence of cytoskeleton fibers. So in order to take into account there's a complexity, we built a few years ago a system to reconstitute what we call artificial membrane-less compartments. And the idea is to be able to control the assembly and the disassembly of such biomolecular condensates with specific physical properties. So we would like to generate actually a liquid-like condensate or a gel-like condensate. And I will show you how actually we use this artificial condensate to study condensate more in a physiological context or in a pathological context. So what are they? What is this methodology? So a few years ago, actually, we start in the lab the design of protein scaffold that actually recapitulate the principle that was highlighted by Mike Rosen. So we need to have a multivalent scaffold, protein that display multivalent interaction, and we try to design actually multivalent protein that can interact with low affinity. So the idea is to use 24-mer, so we use the ferritin core that has self-organized into a nanocage, and we fuse to each of the monomer the mutant of FKBP protein. So this FKBP protein can undergo homodimerization with a typical dissociation constant of micromolar. So the interaction is very weak. So what we expect actually is to have above a critical concentration the formation of a condensed phase. So what we do in the lab is we perform protein engineering. What we put is this construct, we put them inside the plasmid, and we have a M-Cherry reporter. And I will show you here a movie, a confocal movie, where we transfect actually some HeLa cells. And you can observe actually, so there is 2 cell bodies here, and you can observe that within a couple of hours, there is the nucleation of these large bodies, spherical bodies, that nucleate almost randomly within the cytoplasm, and they tend actually to coarsen or to fuse over time. what is interesting with this approach actually is that we have a chemical in the lab that is a binding competitor of the homodimerization domain here. So if we add these small chemicals, we can actually preclude here the interactions. And I'm going to show you. So upon addition of the small chemical, we can trigger the dissolution of these bodies. So it can be very useful for certain kind of applications. So I'm going to just to summarize here the kind of biophysical characterization that we made with these systems and that show that indeed our artificial compartment recapitulate some features of the first order phase transitions and liquid-like behavior. First, we find that indeed we need to reach a critical concentration of protein within the cytosol to nucleate the condensate. Next, what we find actually is that the main pathway towards growth is through coalescence in our case. So you can see here that adjacent compartment can fuse. And what is interesting is that from these movies, if we perform several acquisitions and extract the relevant parameter, we can extract the so-called capillary velocity, that is the ratio between the surface tension and the viscosity. So the surface tension is really the driving force for fusions, and the viscosity will tend to oppose to these fusions. So we can extract this parameter. We also perform what we call fluorescence recovery after photobleaching. So, and we show that the dense protein within the condensate can dynamically exchange with the dilute phase here. So there is some dynamic turnover. Using the electron microscopy, we can actually verify that there is no membrane around our condensate here. And we can also perform some immunostaining of natural occurring condensate in the cytosol, such as the P-bodies or stress granule. And we find that they do not interact non-specifically. So here we were very happy because we were able to reproduce the general features of what we expect to be a phase-separated condensate in living cells. But we wanted after to functionalize these granules in order to answer to some important questions. So I will illustrate this application with two kinds of applications. With first the studies of endogenous RNP, ribonucleoprotein condensate, P-bodies. And at the end of my talk, I will introduce a new project in the lab where we try to test some idea that will examine actually how a condensed phase of protein prone to aggregate, so protein that are linked to specific neurodegenerative disease, can be a driver and can accelerate actually the fibrillation of amyloid-like fibers. So first, P-bodies. So P-bodies are important organelles that are involved in RNA processing, such as polyadenylation, which is important for the global gene expression output of cells. And one striking effect, actually, one striking observation here of P-bodies in red here, so it's an observation of Roy Parker, is that indeed the P-bodies are almost monodisperse and have a small size. So this reminds us a bit what Jean-François introduced. So we don't have, indeed, a large P-bodies in the cytoplasm. So we should expect, indeed, normally, if we consider a simple phase-separated system, through coarsening, having a large bodies. That's not the case. So that's the kind of questions we were interested. So how experimentally in the lab we study the P-body? So the idea was pretty simple. So we start by using our multivalent scaffold, protein scaffold, and we fuse to each of the scaffold an RNA-binding proteins, pumilio, that is highly enriched in the P-bodies. So pumilio, it's a global translational repressor that is known to selectively recruit and interact with 1,000 of different RNA, kind of RNA, for example, messenger RNA, microRNA, or non-coding RNA. So we fuse this RNA binding domain to our artificial scaffold, and we actually try to see if these granules in cell could interact, recruit, or even nucleate endogenous components of the P-body. So, and it works. I'm showing you here the first data that we had on the system. So the artificial compartments are in green here, and we can see actually that it can recruit different class of RNA on the surface. So you can see that each of the red dots here are RNA molecules. So here, for example, it's a non-coding RNA called NORAD that is highly specific of the P bodies and known to be enriched in the transcriptome of P bodies. Here, there is two other examples of messenger RNA, PMILIO2 and RAD21, and this is highly specific. For instance, if we look for RNA that are not part of the P body transcriptome, they do not interact with our artificial compartment here. So the way we actually observe these RNA is by using single molecule fish. So we can even almost count each RNA that are co-segregated on our condensate. So we perform many controls. Here I just show you a control where if we use a non-functionalized artificial granules, we do not interact with the RNA. So generally, an RNA in cells does not come alone. It's covered by many proteins, like RNA binding proteins. And the second experiment that we perform is by looking if our artificial granule is able to co-segregate protein enriched in the P bodies. And here I show you two examples, for example, EDC4 or DDX6. So DDX6 is an helicase enriched in P bodies. And we can even here appreciate actually that the recruitment of DDX6 is on the surface. So it's an electron micrograph. And each gold nanoparticle, black dot here that you have, is an anti-DDX6. And we can see that actually the recruitment is on the surface of our granules. The thing that we observe also here, and that was in a sense surprising, is that we start to observe a size effect. So on neutral granule or non-functional granules, we generally observe 2-3 large aggregates, 2-3 large condensates in cells, whereas here we start to have smaller condensates. And in order to explore more of this effect, we perform an assay where we were able actually to change the stoichiometry within our artificial condensate of PMILIO, the RNA binding protein. So by actually tuning the density, we can observe indeed that we start to have few condensates that have a large diameter. And by increasing PMILIO within our condensate, we have much smaller condensates and a larger number of them. So in a sense here, our working hypothesis was that by tuning PMILIO density within the condensate, we can tune the RNA binding capacity of the condensate. And we have a direct effect on the size. So how size can be achieved, how size control can be achieved. So PMILIO is a rather complex protein. It can really target more than 1,000 of RNA. So in order to test the minimum requirement to have size control, Audrey Cochard in the lab developed, when she arrived as a PhD student, an assay where she was able to build artificial condensate that can really recruit one single RNA species that have a specific size. So in order to do that, she generates condensate with an RNA binding protein, MCP, that can only recognize a specific pattern of RNA. So an MS2 stem loop here. And what she finds first is that RNA is exclusively recruited at the boundary on the surface of the condensate. So you can see here in red, this corona, it's the recruitment of RNA. The second thing that she observed is that upon recruitment on the surface of the RNA, we have a change in the material property of the condensate. The condensate that display liquid-like behavior will now actually undergo towards a liquid to more gel-like behavior. So there is a change in viscosity of the condensate. The thing also that was very interesting with this assay is that using single molecule fish, you can really count the number of RNA per condensate. So you can have actually a very efficient way to link the RNA surface density as a function of the condensate size. So it's pretty unique, and this is something that is not accessible with endogenous condensate. And I will not enter into details. I will just show you 3 pictures here. That shows that at low RNA density, we have large condensate, But when we have an increased RNA density on the surface of the condensate, we have smaller size. So in a sense, the size and the number of the condensate scales with the RNA surface density. So in order to interpret these results, we propose a very simple model where we show that at a low surface RNA density, the coarsening is rather standard. So we have growth of the condensed phase as by subunit additions or by fusions and relaxations. Whereas at high surface density, so we have a change in the viscoelastic properties, but also we may actually preclude or reduce subunit additions, but RNA on the surface can actually act as a sort of physical barrier that introduces steric hindrance and that actually impede relaxations. So that's a very simple model. So it's a passive system. It's not an out of equilibrium system in a sense, like Jean-François told us. So it's a highly timely question. And recently, there was 2 papers that propose an alternative model of size selection for endogenous condensates. So the first one from Géraldine Sédou lab proposed a system where the germs granules are stabilized by a Pickering agent, that is a protein, MEG3, that will adsorb on the surface. And this will allow actually to reduce the surface tension of the condensate. So it's a way to limit the growth of the biomolecular condensate. Another model that I like a lot is from Hiro's lab in Japan, where he studied paraspeckle that are biomolecular condensates present in the nucleus. And what he proposed actually is that paraspeckle size is set actually by an RNA here that has this form, the form of hairpin. And he also proposed a model that is based on micellization, which is not a classical liquid-liquid phase separation model. So it's a kind of interesting viewpoint. And of course, there is a theoretical prediction that shows that size selection can be due to chemical reaction that can be performed within the condensate. So that was what I wanted to discuss about the effect of RNA on the condensate morphology. And I will take the few minutes that I have left to introduce another topics that we start recently in the lab, where we use the same kind of tools, so generating artificial condensate. But in this project, what we try to challenge is the idea that this condensate can be a driver for fibrillation or polymerization of amyloid-like structure. So to do that, actually, we made a collaboration with the group of Ron Melky, that is a specialist of alpha-synuclein, and Leonard Piroska actually started his PhD working on this alpha-synuclein that has its aggregation that is directly linked to Parkinson's disease. So the idea is that during the disease progression, the neurons here actually can accommodate Lewy-like bodies that are accumulation of alpha-synuclein and that display amyloid-like properties. And this leads actually to some cytotoxicity that can have actually important impact for our brain. And what the first experiment and the first design that proposed Leonard was to recreate a condensate enriched in alpha-synuclein. So he adapted the system I introduced earlier, and I am going to show you the main results. So here we can see a HeLa cells, and the alpha-syn is GFP labeled, and you will see that in that case we observe indeed the formation of condensate enriched in alpha-synuclein, and that display the classical morphology, some spherical condensate that appear within the cell cycle, within the cell space. The second experiment that he performed actually was to add the small chemicals I show you at the beginning that allow us to trigger dissolution of the granules. And you will see that within a couple of seconds, we can trigger the dissolution of the alpha-synuclein granules. Finally, he performed some fluorescence recovery after photobleaching, and we observed that the majority of the alpha-synuclein within the condensate actually are dynamically exchanged, And actually, this suggests that our alpha-synuclein condensate display liquid-like behavior, which might be counterintuitive with the idea that alpha-syn normally triggers fibrillations and forms some amyloid-like structure. But the surprise comes later when actually Leonard performed an experimental assay where he had preformed alpha-synuclein fibrils to the HeLa cells expressing our condensate. So Ronald Mackey lab actually is specialist, and they are able actually to generate very well monodispersed and well biochemically defined alpha-syn fibrils in test tube in vitro. And using this assay, we can actually label the fluorescent fibers, so alpha-syn fibers, and we can add these fibrils to our cells. And this mimics actually something that can happen in our brain is when oligomeric fibrils forms within the neurons, and when the neurons actually undergo apoptosis, there is release of the fibrils in the extracellular matrix and diffusion, and this can cause the spreading of the disease. So this assay actually could mimic this infection and spreading of the disease. And what actually we can observe here is the HeLa cells with here the green bodies, it's the alpha-synuclein condensate, and in red here is the fibers, the alpha-syn fibers that we add externally that can actually enter through an endocytic process within cells. And within a couple of hours, the fibrils can escape from the endosomes and can interact with our condensate. So this is here our alpha-syn condensate, and here you can see the fibrils. And within a couple of minutes, what we observe is a morphological transition from a spherical condensate toward a condensate that displays abnormal behavior. So we have really the propagation and the sort of fibrillations of needle-like structures that remind us actually the generation of amyloid-like fibrils. So that's something that was really surprising for us. And maybe you remember the first experiment that has been performed in vitro by Simon Alberti and Tony Hyman. So it's something that is observed in vitro, but here we observe it really in cellular. And the condition is really that we have to have an interaction between the external fibril and our condensate. So our working hypothesis is that upon interaction of the amyloid fibril with our condensate, we have a morphological transition that actually is concomitant with a transition from a liquid to a solid-like behavior. And what we find actually is that there is really, at the molecular level, a change in the properties. And the alpha-synuclein that were in a liquid-like behavior now display amyloid-like properties. So we perform actually some specific biochemical assay to demonstrate that now our condensate display amyloid-like properties. So in summary, I would like to now to sum up a bit. I hope I convince you that we can use these molecular tools and these artificial condensate to study really basic cell biology questions, such as the impact of RNA on the morphology of condensate, and that actually it can be a very interesting platform to study the formation of pathogenic condensates, such as amyloid-like ones. So as a perspective, I just wanted to show you two movies. It will be very short. Two movies where we extend the use of artificial condensate to study their interplay with cytoskeleton fibers. So very often, an endogenous component endogenous condensate can interact with cytoskeleton-like meshwork. And here, Pauline and Audrey, what they perform is they functionalize our condensate with molecular motors. So on the left here, you can see actually the formation of condensate at the vicinity of the cell membrane, and the condensate are functionalized with kinesin-like motors. And we have actually we observe that the active transport trigger the localized nucleation of the condensate here. In this example, what they did actually is that they functionalized the condensate with dynein-like motors. And in that case, we observe on a single condensate that generally form at the vicinity of the centrosomes. So now we are using this system in order to study, for instance, the role of RNA localization in protein productions. And my last slide, actually, is to acknowledge the people that perform the work. So Audrey Cochard performed the studies that allow us to link the RNA surface density with the morphology of our condensate. Leonard Piroska developed the assay to study the role of alpha-synuclein and fibers in order to trigger amyloid-like structures. Marina and Sunichi actually were the two postdocs that developed a few years ago the artificial condensate system to study the P-bodies, and of course our colleagues and collaborators. So Dominique Veil, so she's working on P-bodies, and she knows a lot about RNA containing condensates, and we work together to study the role of Pumilio in the P-body system, and Ronald Melki, that is a specialist of protein prom2 aggregate, and he actually helped us a lot in the project based on alpha-synuclein. Thank you very much for your attention.